Here is Michael Leung.
Thank you. So today I'm going to talk about how to use deep learning to predict alternative splicing patterns. And this is joint work with Hui, Leo, and in Brendan Fry's group at the University of Toronto. All right, so just a customary slide on what alternative splicing is. So it's one of the steps in pre-mRNA processing where one gene can have multiple transcripts. And the most common alternative splicing method is cassette splicing, which is illustrated in this schematic here. So it's the case where you have an alternative exon that is flanked by two constitutive exons, and you can have splicing both ways so that you have two types of transcripts. You can have the alternative exon included or excluded. And alternative splicing is a lot of times measured in something called psi, or pretty much just the percentage of transcripts with the alternative exon spliced in. So in this talk, I'll mention psi to describe this. And in the picture below, I just show that alternative splicing is context dependent. So for example, here in the brain, you could have, let's say 70% of the transcripts that you observe has the alternative exon in it, whereas in maybe heart, you only see 15%. So the goal is to be able to predict alternative splicing patterns. All right, so in our group, we've worked on this for a few years now. It first started with deciphering the splicing code in 2010 by Yusuf Barash, who's in UPenn right now. And in 2011, in the bottom left there, we used something called a Bayesian neural network to significantly improve the predictive capability of the original splicing code, which used boosting. In 2012, our collaborators used a splicing code to look at how the use of features diverge as the evolutionary distance between species increases. So the next thing we wanted to look at is, can we further improve the predictive performance of the splicing code? Well, why do we want to do that? Well, we want to make more accurate prediction that generalizes better so that we can make accurate predictions on unseen data. For example, we're looking at mutations and disease-related features that are related to alternative splicing. And for this talk, I will talk about how we're going to use neural networks to do it, in particular large neural networks. And we're going to show that it's applicable even if the data set is smallish, which is common in biology. Before I do that, I'm going to take just a short detour to talk about what are neural networks, and then with an introduction of deep learning. All right, so artificial neural network, just a brief overview. Here in this schematic here is one of the most common use neural network architecture. It's called feed-forward neural network, where you see connections that are going from the input, X, to the output. And these inputs are nonlinearly transformed by hidden units in the middle there in blue. And you have weights that dictate how these inputs are transformed nonlinearly. And typically, you have your cost function, just typical machine learning, your predictions and target, you penalize it, and your cost function is generally, for neural networks, you could do regression or classification. So how exactly are these inputs transformed into the output? So I'm just going to look at each hidden unit individually. So for hidden unit J here, what I'm illustrating is that all you do is really you take the input, you multiply it by the weights connecting to the input, to the hidden unit, you sum them all up, and then you pass it through a nonlinear function, and that's it. This nonlinear function could be sigmoidal, like the tanh function. Recently, people are also using these ReLU, which is just like a hinge function that implements this nonlinearity. So that's all it is. Now, when people talk about these deep architectures, what all they mean is that you stack a whole bunch of these hidden units, hidden layers together. And I have taken this definition from a deep learning review. So deep learning are machine learning techniques that exploit many layers of nonlinear information processing. And one of the kind of interesting thing about this architecture is that you hope that the machine learning technique transforms your features so that by the last layer, so in hidden layer 3 here, you have very easy inputs, very easy features so that your classifier doesn't have to do a lot of work to do classification or regression. So as an example, maybe your inputs are images, and one of the hidden units has learned what is a cat or what is a human face. And so at the end, it doesn't take a lot of effort to differentiate between these two classes. And usually with these deep neural networks, at the end, all you do is you tag on a logistic regression, and that's pretty much it. So, I mean, these deep architectures, they've conceived a long time ago. A lot of work was done at U of T. But the harder question is, how do you train them? And this is recent developments has enabled us to train neural networks. So I have this point here. So why the interest in neural networks again? I mean, they were popular in the 1980s, but then they got took over by SVMs. They're interesting again because now we have increased computational power, and we have much lower costs of hardware, in particular GPUs, and they've become very important to deep learning, as well as many advances in machine learning research, many of them being in initialization techniques, how to properly regularize them, different optimization techniques. So now we can train deep neural networks. So why do we care? The main thing is they've been largely successful in various domains, such as in computer vision, natural language, and audio processing. And they've really beaten benchmarks that have, so previous benchmark might have improvements of 0.5% or 1% every year. With these emergence of deep learning, we've seen improvements of 10, 15% within a single year. So they're pretty significant. And just to give it a little bit more credibility to deep learning, who is using them? It's Microsoft Research, Google, IBM Research. Recently, Baidu from China is using them, and Facebook in 2013. And you actually see them in a lot of consumer products. For example, if you have an Android phone, you can talk to it, and it'll recognize what you're saying and do search. That's powered by deep learning. What's coming from Microsoft and Skype is that you'll get real-time translation, so you can speak to it, and it'll translate it to a different language. So they're very powerful techniques. All right, so computational biology-wise, can we use these techniques that were developed from the deep learning community? Well, why do we care? Well, after all, biology is quite complex, and we'll need bigger models to appropriately describe it. And so we ask, is it applicable to the kind of small data set that we're used to? I'll just reference this example here, and that's from 2012 from U of T, where a team using deep learning and GPUs were able to win first place in a molecular activity challenge from Kaggle without much knowledge in the biology. And furthermore, deep learning can potentially give us better predictive capability. Potentially, it might not work for all the data, but all the other problems, but it should work good in some models. And with the rapid growth of omic data, I think we do need methods that scale well with data volume, and these deep architectures with GPUs can really scale with data. And last, I just want to talk about this feature learning aspect of deep learning. So a lot of the work that is done, we have to craft our own engineered features to feed into our classifiers. A lot of times now in deep learning, you just feed them, for example, raw data, raw images, and you let the algorithm itself figure out what are the best features to use. So for this work, I'm still using hand-engineered features, but there's some, just want to talk about the potential for just the future using raw RNA, just raw DNA sequences to do our predictions. All right, so that's it for the neural network and deep learning. So we're going to talk about what we did. This data set that we're using is it's an RNA-seq data set in mouse for 5 tissues, and it's from Keisman's group. We're studying 11,000 cassette exons. Input in the model is the tissue type. We want a tissue-specific splicing code. And we also have genomic features that are extracted from the genomic sequences that are position-specific. And some of them are listed here. The target that we want to predict is psi, as well as the change in size. So we want to predict delta psi between different tissues. And here is the deep neural network that's used for the prediction. So on the left here, you see the input being the genomic features. In the first hidden layer, it actually shrinks the dimension of the features. And then we tag on 2 tissues as input. So we have 5 tissues, and we use a 1-of-n encoding. So there's 10 inputs into the model. And we pass it through 2 hidden layers. The numbers there indicate how many hidden units are in the model. And we predict 2 things. First, for each of the 2 tissue, we predict the absolute size. So whether it's going to be low, medium, or high. Low being 0 to 33%, medium being 33 to 66, and high being 66 to 100% psi. We also predict the tissue difference. So for these 2 given input tissues, what is the difference between, what is the psi between them? So we also phrase this as a classification problem. If the delta psi between them is less than 15%, we say it's no change. Otherwise, it's either increase or decrease. And I think you'll find that maybe this is kind of a brute force way to do it. We're multitasking, we're training multiple tissues at the same time, we have multiple tasks. But I think it's really a way of saying we're going to give the model full flexibility and just let the data tell us what to do. And for the remainder of this talk, I'll describe this code as the low, medium, high code, and the DNI, decrease, no change, increase code. And you might wonder, why do we even need the DNI code? Because yes, you can just use the low, medium, high code and look at the difference between the splicing pattern there for the 2 tissues. But we find that it improves our performance a little bit, and so we included it in this model. All right, so just one slide on training the model. The details are in the paper. We use stochastic gradient descent with momentum. One of the important things we have to do is you use these balanced mini batches. That is, you have to make sure as you train, when you do your gradient update, so that you're sampling from the training data so that the distribution is even. So for example, you want to make sure that you have the equal number of tissues for each mini batches. And then we use dropout regularization. This is a new regularization technique that has been quite popular in deep learning architecture. And we also did hyperparameter search using validation data. And one of the important things about this is you definitely have to use a GPU to do this. With GPU acceleration, we've decreased the training time by up to 15-fold. So instead of training your model for 2 weeks, we can do it in 1 day. And this is very important for us to be able to do a hyperparameter search. It's also interesting from the hyperparameter search that a lot of these numbers, they were not chosen by me. They were chosen based on validation data. And these very high number of hidden units actually was unexpected, because initially I used much smaller number of hidden units of the order of 1,000 here. Another interesting thing is that our genomic features are actually quite rich. If we go below, say, 150 to 200 in this dimensionality reduction, we actually get a significant decrease in the performance of the code. So we do need the number of hidden units, the capacity of the model. And finally, we just do the standard 5-fold cross-validation. We use 3 for training, 1 for validation to do the hyperparameter search, and 1 held out for testing. So this is data that we've never seen before, and all the results I report are based on test data. All right, so the architectures I've used in comparison is this Bayesian neural network from our previous work, deep neural network, and just a logistic regression, just as a baseline. All right, so the results, I'll first talk about the low, medium, high code for the 5 different tissues. You can pretty much ignore the black, the MLR logistic regression, and just look at the DNN and the BNN. You can see that it especially excels in the medium category. So the 3 columns says, so for brain, we say brain low, brain medium, and brain high. And it's interesting that most of the improvement are actually in the medium category, because these are actually the cases where you have alternative splicing differences between tissues. And now that's why we decided to look at the tissue-specific exons. So these are what we believe to be actually hard examples to predict. And you can see huge improvements. You can see that the white bars are always statistically higher than the gray bar. So it performs quite a bit better, especially in determining tissue-specific splicing. So why are we looking at tissue-specific splicing exon at all? Because we claim that these are the hard examples. For example, you build models. Let's say you get it right 80% of the time, but there are 20% of the cases that you're not doing good prediction on. So you ask, is your model actually well? Because it's not actually accounting for these difficult cases. It's not clearly capturing the entire story. But if your model has enough capacity, as shown here, you can do well in the hard cases as well as the easy cases. So I think the take-home message is kind of the capacity of the model does make a difference. And if you look at the DNI code, you look at how well the code is able to perform by differentiating between psi for different tissues. I don't even bother showing the logistic regression here. The DNN clearly excels across the board. So next we ask, can the model generalize across a different data set? So we trained it on this data set from Keisman's lab. So this is this code performance on a different RNA-seq data set. So these are results from a different group, different research group. And just focus on the white bar and the gray bar here. Sorry, actually focus on all the bars. The black bar is the original work, Bayesian neural network trained on the Keisman group data. DNN, the gray one, is just repeating what the results I've shown in previous slide. The white here is the interesting thing, where we've used our model trained on the Keisman, the Brawan data, and used it to predict what happens in a different group from Benjamin Blanco's lab, the Barbosa-Moraes data. And if you look at the heart, you can actually see that the model performs pretty much equally well for the heart in both the low-Miyahai code and the tissue-specific cases. For the brain, you actually see some decrease in performance, but that's I think expected because the brain is naturally a very heterogeneous tissue, and so we expect that we do less well. And the same story in the decrease no change increase code. You see the white having a drop in performance. We can still see that it's still on a different data set from a different lab. We still perform better than the original splicing code. So our model generalized as well. So the final part of the presentation is, this neural network, they're often criticized being black boxes. Can we actually interpret the neural network? So there's some options. You can look at the weights, but that's sometimes not very useful, especially when you have data that's not very structured. So for images, you can actually look at the first layer weights, and you can see some strokes, you see some patterns. So for this, it's not so good. You can evaluate how each of the input changes the model performance. That's easy to do. And you can also evaluate the feature sensitivity. So you calculate del y, del x, and you can look at how changing each input changes the output. But I say that the criticism is that you're looking at each input individually, and so you might not be able to account for interactions between the inputs. So there's a recently reported work called using the class saliency to look at what the model is doing. So from the trained model, for each example, what you do is you forward propagate using that example from your data to get the output. Now you perturb the target. So let's say the model predicted low. You're going to say, all right, I want you to predict medium, or how you perturb it. And then you do back propagation. Now instead of updating the weights, you back propagating all the way to the inputs. So you ask the question, what do I need to change the input to make a different prediction? And the magnitude will tell you that if the magnitude is high, it's saying this input only needs to change a little bit to affect the output a lot. So that's what we did. And you can do something, you can produce charts like these. So you do this for all the data set, all the data that you have, and then because we're doing gradients, we actually get the direction of change as well. So we get a table of saying how each feature changes the psi. Actually, I need to make a clarification. In the proceeding, there's an error. It should actually say silencers instead of enhancers. That's a typo. We'll get it corrected from the journal. Now we also talk about we're talking about tissue-specific splicing code. So now we want to look at the sensitivity for how different tissues uses the features differently. And so we have this table here. On the left is the different the tissues and how bright the box is. Also, the x-axis shows the feature that we're using. And you can see how each tissue is actually using the tissues differently so that we can make tissue-specific predictions. And you can see that the kidney and liver actually are quite similar in terms of in this table, and it's true that our model actually has a more difficult time predicting kidney versus liver psi. All right, so the performance improvement. So I claim that because we have a large model with sufficient capacity, and this is just my guess, is that we have these regulatory programs that are potentially multimodal. And if you have a simple model, so you might only capture a part of the regulatory program, but not all of it. And I think with good regularization, good optimization, we're able to train these models with sufficient capacity to address a different part of the regulatory program. And yeah, there's a better optimization regularization technique as well as the GPU-enabled hyperparameter search. All right, so that's the conclusion. I've talked about a splicing code with improved predictive performance. We demonstrate that deep architectures can be beneficial even on a relatively small biological data set. And I've shown how you can analyze a model in terms of its input features. Now, future work, our lab is interested in looking at other post-transcriptional modifications. We're interested in actually learning directly from the genomic sequence themselves instead of crafting features. And yeah, more tissue, more species, more data. And these are the people involved: Hoi with the Bayesian neural network, Leo in the middle with the feature extraction pipeline, and my supervisor, Brendan. And these are the funding agencies. Okay, thank you.
Thank you. Any questions? If you have questions, please step to the middle to the microphone.
Hi, thank you for the talk. I mean, how did you select these 500 or whatever initial input features? This is the first question. And the second question, ultimately, those transcripts should become proteins, right? Or isoforms or something. Did you take into account the probability of this isoform to become a protein or to make any?
Okay, so I guess your first question is on the features. Yeah, that's a pipeline that we've been developed over many years, where we scan the literature to look at what's relevant for alternate splicing that we've learned over the years.
And you put everything that you found?
We put everything that we found, all 14,000 features. The other question is, no, we're not looking at isoforms. We're only looking at RNA-seq data. We look at the number of mappings in the junction. So we're counting transcripts. We have a way of processing it. But yeah, basically counting transcripts. Yeah.
Okay, it was a great presentation.
Thank you.
So in some scenario, you test the model, right? Like this is brain, this is muscle.
Yeah.
You can train it, okay? Then at the last moment, you figured out, the muscle wasn't the muscle. It's kidney.
Yep.
Our machine, this wasn't the muscle. It's kidney. What happened? So you got a totally wrong training. So in the final step, you figured out that was wrong, and you want to roll back and retrain them.
Well, I guess I don't know about retraining, but there's got to be noise in the data. So you use what you have from the data, and you do the best out of it. I mean, I think it's a common problem for any kind of machine learning problem. You can have corrupt data.
Yeah, as far as I understand, the deep learning, what is different from the ordinary learning is that they could handle some kind of more complicated situations like that, like a paradox situation, something like that. So you titled deep learning, and I just wonder, in some...
I don't know about actually switching the labels for your data, but I think I do agree that it should be able to account for more complicated regulatory programs.
Mistakes, some kind of.
Yeah, mistakes, yeah. Yeah. I don't know.
Thank you.
Okay.
I have a question regarding your pre-training of the data. Have you used some kind of pre-training methods like restricted Boltzmann machines or stacked?
We just use an autoencoder.
Yeah, but you can use them for tree training and then afterwards put just the last layer for the classification on it.
Yeah, we could do that, but we chose autoencoders, which are commonly used as well for pre-training. So yeah, it's easier to do for me, I think. Yeah.
Hi. It was a really great talk. My question is actually pretty. It's just a quick question. So you have in your results you have compared basically different machine learning algorithms to each other with the deep neural network.
Yeah.
have you tried any method that's not basically machine learning based, more like kind of MISO, which or COFDIT that actually tries to differentiate, basically tries to find the splicing between two different samples?
Sorry, I missed the, what should I use instead of machine learning algorithms?
So I was kind of wondering whether if you actually tried comparing the machine learning based method or just the deep neural network to other methods that that is more statistical, like MISO or COFDIF. Which are
Not
available
those
to
methods that
compare
you
to.
mentioned, but in our original work, we did compare against regression, logistic regression, SVMs. We did a couple of methods and shown that the Bayesian neural network does better. And now we're showing that the deep learning method does even better than the BNN.
Okay.
So not in those methods that you described, but we
see.
did do some benchmarks.
Okay, thank you.
I have a question about the architecture of the neural net. So you
Yeah.
have this feature that connect the first layer of the hidden units,
That's
right?
right,
And then
yeah.
you have the tissue type that connect to actually the second layer of
Yeah.
hidden units. What's the rationale behind that?
So the first layer is kind of to reduce the dimensionality of the input feature space, which is quite large. It's 1400.
Right.
And there are also duplicate features, or features that can mean very same thing, very similar, with
So you
very
use,
similar
like,
definition.
One of K encoding, right? So
Yeah, so then
Yeah.
we reduce that feature space so that one of n encoding doesn't get overwhelmed by the large input features.
Okay.
Yeah.
And then when you actually do the feature selection, you say you perturb the target label, and then you will change the features in order to match with the perturb label. Is that how you work?
The perturb part is to
Yeah.
understand what the network is doing. So you do forward propagation,
Yeah.
change the output, and then do backprop all the way to the input, not the tissues. We don't care about the tissues,
Okay.
to understand the features.
Okay, and then you rank the feature based on the changes.
And then those changes tell us which feature is important.
Okay, I see.
Yeah.
Okay, thanks.
Yeah.
Hello. I'm curious about how do you initialize the number of layers in an unit?
That's part of the hyperparameter search that we ran based on validation data. So those large, so I think you can read the paper for the details, but there's a automatic hyperparameter search framework that's been developed also at U of T that helps us.
Okay.
Yeah.
And another question, I'm a little bit confused that what do you mean it's a tissue specific method, and do you mean for each tissue you build a deep learning network, or
Oh no,
the
so
tissue is
there's
a
only
kind
one.
of feature in your input?
Oh, so yeah, what I mean is there's only one model ever for this whole work that can predict everything. What I mean by tissue specific is that those particular exons that exhibit tissue specific splicing. So I have 5 tissues. Those examples where there's some high psi in one tissue and low psi in other tissues, those are the cases where there's tissue specificity. Those are the harder cases to predict. Most of the data set is actually tissue constitutive splicing, so where it's always low in one tissue and always low, it's always low in all the tissues for one exon and always high for all the tissue and another exon. So that's about 80% of the data. About 15-20% of the data are these more tricky cases that we're able to do especially well compared to the previous work.
Okay, thank you very much. Let's thank the speaker again.